Alcohol fermentation, also known as ethanol fermentation, is a biological process in which sugars such as glucose, fructose, and sucrose are converted into cellular energy, and it produces ethanol and carbon dioxide as metabolic waste products because yeast performs this conversion in the absence of oxygen-ethanol fermentation, and this is classified as anaerobic. The equation for alcohol fermentation is C6H12O6 yields with C2H5OH plus CO2.
The process of alcohol fermentation can be divided into two parts. The yeast breaks down glucose to form two pyruvate molecules. This part is known as glycolysis. Second part, the two pyruvate molecules will be converted into two carbon dioxide molecules and two molecules of ethanol known as alcohol. This is called fermentation. The main purpose of alcohol fermentation is to produce ATP, the energy currency for cells, under anaerobic conditions.
Let's take a look at the process of alcoholic fermentation. As you know, it begins with a molecule of glucose. Chemical reactions cause the glucose molecule to split into two. This results in two molecules of pyruvate, as you can see. This chemical pathway yields a molecule of ATP here and yields a molecule of ATP here. This is very important, energy carrier, and this is what we're after, so we will flag that with dollar signs. The oxidation of glucose doesn't happen by itself, but it is rather aided by the power of a molecule called NAD+. And in the process, it is converted into NADH. What I'm showing you above also happens here, so I'm just going to say same, so you'll understand that this pathway is just like the one above. Next, the pyruvate loses a molecule of CO2, this is a gas, and turns into an intermediate called acetaldehyde. Now the goal is to make lots of ATP. In order to do that, we need to split a lot of glucose. And that's only possible if we have a big supply of NAD+. So the only way to obtain a big supply of NAD+ is to recycle the NADH back into NAD+. That way we can use it again and again and again and split as much sugar as we want. And the only way to do that is to convert the acetaldehyde into ethanol, which is toxic. It's a waste product that must be eliminated or degraded by the chemical reactions of the cell. The saying, Leo the lion says grr. Leo stands for lose electrons, oxidize. And grr says gain electrons, reduce. In this model, NAD+ is gaining an electron from the glucose. So NAD+ is being reduced. And that's how it turns into the neutral NADH. And of course that means the glucose is being oxidized.
Alcohol fermentation can be used as fuels, preservatives, solvents. Because ethanol can be used as a solvent in medical drugs, perfumes, and vegetable essences such as vanilla. Another is alcoholic beverages, antifreeze, antiseptics, is used to disinfect the skin.
Product recovery. It can also be recovered by fractional distillation. Distillates 95.6% alcohol and 4.4% water. Alcohol is obtained by distillation and column is known as rectified column.
The alcohol production is an important fermentation because of the following reasons. It gives ethyl alcohol used for consumption as well as industrial purposes. It gives byproducts like carbon dioxide, yeast biomass, fertilizers, and etc. Gives effective utilization of agro-waste, molasses, sulfites, waste liquor, and etc.